Ladies and gentlemen, thank you for your patience as we get started here. I would like to welcome you this evening to our live broadcast from the Templeton High School Performing Arts Center. We're here tonight to celebrate the 7th annual STEM Symposium and 7 amazing scholars that you'll see before you here today. These 7 students have really risen above in the fields of science, technology, engineering, and mathematics. They have completed year-long internships or senior projects that really help them stand out in their science fields. I think that as you follow through tonight, you will be as amazed and as proud as I am at watching the progress and the things that these students have learned throughout these 4 years. So I am glad that you could join us this evening. We are going to be starting this evening off with certainly a region that was most well, let's say not exactly a quiet area to be working during the pandemic. We'll be kicking it off with our interns here tonight, and we have Alex Harding from our molecular sciences strand coming to tell you about his experiences. Please welcome Alex.
Hi, everyone. Hi everyone, my name is Alex. I'm part of the molecular sciences strand, and I did my internship at the St. Louis Obispo Public Health Laboratory. It's right off Johnson and Slow, and it's directed by Dr. James Beebe. Some of the stuff I did during my internship, unfortunately, this is the only 2 pictures I could take. Gloved hands with bacteria on them, and phones don't mesh super well. So these are 2 rare photos I was able to take. On the right of your screen, you'll see a bunch of tubes in a hood. That's where I spent probably 20 hours just pipetting liquids back and forth. And it seems really boring, and it was, but it's really important because what that is it's a media where the scientists would take the coronavirus swabs and put them in the liquid, and it would basically remove the skin of the virus so we could access the RNA inside and test for it. So it was very important for the pandemic efforts. It's certainly a very new technology. On the left there, you'll see what we call a Quanta Tray. And so what it is we collect water from the ocean all up and down the coast, and we test it for E. coli, total coliforms, and enterobacteria. And so this one, I believe, is total coliforms. I don't remember exactly. They're pretty similar. But the yellow squares are positives. And so it's just a media in the liquid that the bacteria reacts with to make that yellow color. And then when you put it under a black light, if it fluoresces, then it's positive for E. coli. Now, next I have some pictures of stuff that I observed there. Obviously, these aren't mine. I couldn't take most of these pictures, but I think they're pretty interesting. So here we go. This is not a picture of space. What this actually is, it's a slide of rabies that is dyed with a fluorescent dye. So when you put it under a black light, it fluoresces. So those slightly brighter clumps, that's where there's a higher concentration of the rabies virus in the brain tissue that we get from animals. And those are most often given to by animal service. We tend to get foxes, raccoons, bats, skunks. We've had a horse once, so that was exciting. And this is a pretty rare thing. We'll get something about once every 2 months, 1 to 2 months. And unfortunately, I can't observe it. I'm not vaccinated for rabies, but if I was, I would love to. Next up, we have what's called a TSI slant. TSI stands for triple sugar iron. I don't remember what the sugars are. It's like glucose, sucrose, and fructose, I think. And then there's iron in it. And all it does is you put a bacteria on that slanted part, and it eats some of the sugars, and that's what the color changes are. So if a bacteria eats just fructose, then it would change, say, the top part red or something. And then the iron, it reacts with an enzyme in bacteria to form hydrogen sulfide, and so it'll create kind of black, bubbly liquid towards the bottom of the tube. This is a very common test that we do in the laboratory to test for a wide range of bacteria. It's a very good test to kind of narrow down what you're looking at. So it's a pretty common test. I've observed it quite a few times. Next up is a picture of a parasite called Gerardia Landlia. I like him a lot because he looks like he's smiling, so I think he's kind of fun. But he's typically found in water, that's in untreated water, so like well water most commonly. Then also can affect cats and dogs. And if it comes to the public health lab, it can affect a human as well. This is pretty rare. We don't see these too often. I've only seen preserved slides of these, but every now and again you'll see one. They have pretty mild effects, but they're enough where we have to look out for them. Next up, I have a picture of a microscopic view of a mold called Aspergillus fumigatus. This is one of the more common mold infections you can get. It causes symptoms similar to that of the flu, or in bad cases, even pneumonia. And it is a deep respiratory infection. So you inhale it and it gets into your lungs and just kind of grows. So if it's left untreated, it can be lethal. However, with antifungals, it can be pretty easily treated. And finally, we have a picture of Streptococcus Pyogenes. This is a very common bacteria, kind of like E. coli. It lives basically everywhere, and it's really hard to kill without like bleach. But those clear rings you see around it, we call this cell beta hemolytic. So it'll eat the blood in the red plate there, that red gel, and for nutrients. And I think this is just kind of cool because you don't think of bacteria doing this sort of really weird vampiric almost act. But these blood agar plates are really common as well, because about half of common bacteria are either beta hemolytic or non-beta hemolytic. So it's a really good way, like the TSI slant, to just kind of cut out half the bacteria you're trying to find. So next, I'm talking about what's next for me in my science career. I will be attending the University of Utah, and I will be majoring in laboratory sciences in their medical school there. I'm taking a lot of classes, a lot of labs, like in parasitology, mycology, virology, and microbiology. And this is basically what I was doing at the laboratory. So I think it's a really cool opportunity for me, especially. And then I plan to further that by going into medical school to study pathology. Finally, I'd like to thank Dr. James Beebe, Stacy Evans, the two directors of the laboratory I was at, and then Elizabeth Farrington, my supervisor, for kind of showing me how to do everything, which is very helpful in a government laboratory. So that was my presentation. Any questions at all? If you could write them in the chat, that'd be great.
Ladies and gentlemen, if you do have any questions, please feel free to go ahead and type into the chat box right there. Well, where are you going? Type them on into the chat box, or if you would like to unmute, you're welcome to go ahead and unmute to ask questions as well.
That's very small.
It's the first question here was, hold on, wait for it. Why do fish swim in salt water?
That's my dad.
Is that your dad?
Yes.
Because the pepper water makes them sneeze. Does anyone have any questions for Alex about his internship? Okay, please give Alex a round of applause. Thank you so much, Alex. Great presentation. So continuing on with our interns here today, we are going to welcome up next Julianna Jarvo, who completed her internship in molecular sciences. And what, might you ask, do chemical compounds and computer programming have to do together? Well, Julie's going to tell you all about it. Come on up, Julie.
Hello, my name is Julianna Jarvo. I did my internship in molecular sciences. I actually got my internship at the beginning of this summer. It was kind of the height of COVID, so it was hard to find a place where I could be in a lab for my molecular science internship. But I was able to find an internship with Dr. David Ziegler at Cal Poly, who's a chemistry professor. And I was able to work remotely doing computer programming for his data that he had from chemical equations that had been generated, I was able to write computer code to parse that data and to make graphs and models of electrical and chemical models for that. Here's some of the code that I worked on, and here's my workstation. Dr. Ziegler was accommodating enough to send me a computer, as you can see in this picture, that ran Linux because my computer at home did not. So that was very helpful to get me started in this area. I worked on two main projects. One of them was making Gaussian curves, so graphs for the excited state energies of certain molecules under different inputs to their environment. And my other was making models of orbitals for certain molecules, and I generated a lot of data. It took me 3 days on my computer to generate all of the data from the file that had been building up at the chemistry lab that they had been waiting to parse because it took so long to parse it by hand. One of the biggest impacts that this had on me was learning how to work through my failures. As you can see, here's a piece of computer code that has errored out multiple times. But through this internship, I was able to, even though sometimes I felt like giving up, it was the middle of the night, I had been working for hours and one simple thing wouldn't work. And even when I fixed another thing, there were just more errors. But I was able to work through them, and I was able to find out when I could fix it myself and when I needed to ask for help and find that balance. I was able to learn that I did have what it takes to work in the computer science field, and it was possible for me to get through those errors, especially just setting up equipment for a chemistry internship when I hadn't been working with many of the software before, but I was familiar with it by the end. Here are some of the molecular or electron orbitals of certain molecules that I generated, a few of thousands. And during my internship, it wasn't just me and Dr. Ziegler. There were also a few undergraduates who worked with us. And it was really nice to be able to see them, to be able to work with them. There was a collaborative environment between us, but we all had our own separate projects. So it was good to see everyone's projects come together at the end, even though it was more of an independent work environment. So I was able to work on my own. I knew there were always people there to help me, and I really enjoyed that scientific environment of collaboration. And everyone who I worked with was very into chemistry. They very much enjoyed it. They looked forward to that and saw that in their future. So I'm not sure if I, after that, want to go into the chemistry path as much, but I would love to go into biology or some other combination of computer models, data science, and other forms of science. One of the most exciting things that happened to me during this internship was I became a co-author of a research paper with Dr. Ziegler and some other people who were doing the research at the lab. Here's the abstract. At the bottom, you can see one of the Gaussian curves that I graphed using my code. And this paper was submitted to the Journal of Physical Chemistry in January, and hopefully it's submitted for review. It's being under review, and hopefully it will be published soon. That's a big accomplishment for me to have in high school. And I was glad my work got to come together with all these other great researchers to have that finished product. After this internship, as I said, I'm excited to go into the computer science industry. I'm hoping to major in computer science and go into computer science and data science fields in the future. I'm planning on going to Columbia University and with that computer science major. Thank you. Are there any questions?
Thank you, Julie. Does anyone have any questions for Julie? I've got another set up here. I think we'll be a little bit better for questions right now. Any questions though? Computer engineering and computer programming to help better understand chemical compounds. Definitely a phenomenal field and something that we can do so much with in the future. Okay, thank you very much, Julie. Coming up next, we have stairs only on one side. Coming up next, we have someone who has been doing her internship, well, for a while now. It was an AP research project that merged into something that is a career that she is fascinated to follow through with. So I'd like to welcome up Lauren Allen. Come on up.
My name is Lauren Allen, and I am in the Chemical Engineering STEM program. I have been immersed in the California wine industry my entire life. My grandfather has made wine for 50 years, and my mom is the owner of a vineyard and winery consulting business. I want to become an integral part of the wine industry's future and carry on the family legacy. Last year in my AP research class, I began my journey in winemaking by conducting an experiment and writing a research paper on CRISPR. What is CRISPR? CRISPR uses a guide RNA to guide a Cas9 enzyme known by many scientists as molecular scissors to a desired location and then unzips the double-stranded DNA. This is where you can introduce changes such as an insertion or deletion of a gene because the cell will start to repair and replicate that change. I gene edited wine yeast and analyzed the fermentation progression of Grenache Blanc and Albarino varieties. I chose this topic out of curiosity. I was unaware of the inner workings of CRISPR, and I wanted to envision an experiment that was unique to the exponential growth of CRISPR innovation. I thought to myself, why not mesh this new biotechnology with the expanding Central Coast wine industry? My study ended up reflecting on how Grenache Blanc and Albarino fermentation with the deletion of the ADH1 gene and insertion of the GFP gene marker in X-Pure Zemaflor wine yeast can affect fermentation progression and alcohol levels. I started my procedure by harvesting and crushing grapes in my pajamas. Yes, very embarrassing, but it was really early in the morning, to utilizing the Odin bacterial CRISPR and fluorescent yeast combo kit for most of my materials, and reading multiple scholarly articles on fermentation progression, yeast functions, and many CRISPR procedures in order to create and customize my own Frankenstein wine. All of my results have justified the hypothesis I proposed. Using CRISPR to delete the ADH1 gene does in fact inhibit wine yeast to properly convert glucose fructose to ethanol. This outcome creates a wine with a lower alcoholic level. The GFP gene marker was also very successful in turning the yeast a fluorescent green color under the presence of blue light. The graph depicts how the ADH1 and GFP have similar conversion rates. The gene-edited yeast on the Petri dishes had a slower growth. This growth pattern translated to similar behaviors during the fermentation. The control yeast were more vigorous because they had a functioning ADH1 gene. There are numerous factors that contribute to why the Albarino was not as successful as the Grenache Blanc, but as an observed fermentation in my study, it was apparent that the Albarino and Grenache Blanc were very inactive compared to the yeast in the controls. My results showcase how winemaking and gene editing have the potential to become interrelated as CRISPR technology progresses. After my research project, I was very inspired to pursue an interest in biotechnology and winemaking. With my new learned experience and desire for more experience, I acquired a lab internship at Dow Winery. I am very fortunate to find a job where I get to work alongside experienced chemists and winemakers. Biochemist Katie Ayers, chemist Mikey Mott, both graduates from Cal Poly, and owner Daniel Dow have taught me everything I need to know about being successful in the lab. They have created a positive and productive environment that has allowed me to find my interests and create accurate data. There are multiple aspects to wine analysis in the Dow lab. I have learned how to fix and operate analysis machines, such as the Gallery, the Metler, and the Alkalizer, as you see on this slide. I've also learned how to prepare samples, interpret data, inoculate wines, measure out additives and solutions, and countless other valuable job skills that I am fortunate to have learned. I am fortunate to experience the whole process from cluster samples to harvest full chem analysis panels, and finally to bottling the wine. I feel like I've gained the most insight into my field from harvest, the busiest time of the year. During harvest, I experienced the pressure of fast and accurate results while also making split-second decisions that determine the outcome of the wine. I remember one day I had tons of phenolic samples to do. The phenolics machine is a UV visible spectrophotometer that creates a graph, as you can see in the picture on the slide, and tells you the different chemical compounds related in the wine related to color and taste. That day, Daniel Dow and some executive marketing members were in the lab looking over the phenolics numbers. I immediately noticed that the phenolics numbers were not looking right. Instead of panicking, I tried to stay calm and demonstrate my problem-solving skills. I recalibrated the phenolics machine, and the baseline test was still incorrect. I realized that the previous sample was not put into the centrifuge, and sediment from that sample was sucked up into the tubing, which skewed the data and clogged the entire system. I quickly replaced all of the tubing, and it finally worked. This experience was very nerve-wracking, but I realized I made a mistake and fixed what was wrong. I feel like I handled this situation professionally, and Daniel Dow and his guests were very understanding. I learned the power of maintaining accurate data and how being educated on the procedures and the machines you're working with is key to being successful in the lab. I felt very accomplished that I helped produce great wines during the 2020 Dow harvest. Now that I have an enhanced understanding of winemaking and continue to learn, I feel like my possibilities for furthering my wine research are endless at Cal Poly. I will be studying viticulture and enology with a concentration in enology. I hope to integrate CRISPR and other biotechnologies into my studies at Cal Poly as well. My end goal is to become a sommelier, winemaker, and owner of my own wine analysis lab. I've always set hard goals for myself to achieve, but strive to accomplish them to the best of my abilities and am dedicated to making it happen. I want to make my grandfather and my mom proud by pursuing a unique path and becoming successful in the wine industry. Thank you. Any questions?
Does anyone have any questions for Lauren? Let's see. The wine industry could use more solid chemists.
Yes,
Good luck at Columbia. Absolutely. It's been such a joy too. Lauren was in my AP research class last year in starting this whole process right here, and to see her grow in her skills and accomplishment in the field has been phenomenal. So
Thank you so much for this
Absolutely.
opportunity.
you're very welcome. I'm very lucky to get a chance to work with such amazing scholars, and I hope you guys at home are getting a chance to see just how wonderful an opportunity this is for us as educators too, to work with such wonderful, hardworking students. So coming up next, we're going to segue from the world of wine to the world of music. Coming up here, what do music and math have to do with each other? Well, a whole lot. I welcome Elijah Esparza coming on over to tell you a little bit more about it.
Hi, so for my research project, I analyzed the changes of bass guitar frequency spectra caused by overdrive pedals. Over the summer, I had an idea that I was going to have to research something math-based. So over the course of the summer, I kind of found some inspiration in the Fourier Transform, which I'll explain in a little bit. I then came across two potential research topics, an equalizer and the fast Fourier Transform algorithm. Both of these were very interesting topics, but ultimately not what I could research in a year with my current knowledge and limitations of the research paper. So from there, I found an interest in overdrive pedals. I play music, I play bass a little bit, and I found an interest in maybe some frequency analysis or some hardware analysis of overdrive pedals. About a month into the school year, I ultimately just decided on the frequency analysis, both as a time and a data sensitive choice because of the limitations of only having 1 year and a 5,000 word paper. So I ultimately landed on this research question. From a mathematical perspective, how do guitar overdrive pedals differ from bass overdrive pedals in terms of frequency and harmonics, and what qualities optimize the sound and appeal of a bass overdrive pedal? This took quite a few steps to research, 4 distinct multi-step stages I took to solve this question. I began with learning the mathematics, initially Fourier Series, which is very related to the Fourier Transform. Fourier Series is the concept that you can use an infinite number of sinusoids, like a sine curve or a cosine curve, to add up to any other periodic function. As you can see on the screen there, that square curve can be ultimately created with enough sine curves added to it. And I left the equations there so that you can see the similarities to the Fourier Transform. They're very similar concepts, just the Fourier series maybe from like the front, whereas the Fourier Transform is almost like from the back. With the Fourier Transform, you already have that final function and you're breaking it into the different components that make it up. I really like that picture because it really shows how the Fourier Transform works. You have that final function that's made up of 2 other functions. And on the frequency domain, you can see the different amplitudes and different frequencies that exist in that function. Once I had grasp of the math, I implemented it into a Java program. I started with a generic primitive discrete Fourier transform, which is just like the regular Fourier transform, with generic sample data that I found online, just a simple 20 Hertz sine curve. From there, I realized that I was not going to be able to get through all of the data I was going to need with this generic discrete Fourier transform. As you can see on that graph on the bottom of your screen, the very quickly increasing curve is the fast Fourier or the discrete Fourier transform in terms of processing time and number of calculations needed, whereas the one that's barely above the x-axis is the fast Fourier transform. The fast Fourier transform is an optimized version of the Fourier transform and a lot more efficient, and therefore what I was going to need for the amount of data I was going to have. With most of a program written, I began recording. I used a bass guitar that I already owned and played 7 different notes, all of the natural notes. As you can see, all of the dots and squares on the image on the top display what notes I played. And I used the bass clean and recorded that in GarageBand first. And then I used the 4 overdrive pedals that you can see below, the Boss ODB3, the Aguilar Agro, the Boss DS1, and Orange Burst. Each of those I used different settings to get a variety of sound and kind of optimize their potentials, and record all of those samples into GarageBand, 112 samples between the different notes and different pedals and different settings, and put those all into GarageBand, clip the audio down to get a shorter amount of data, and then changed it into a text file that I could then use for graphing. I ended up with about 10 million data points with all of the samples having 44,100 data points each, and then having 112 of those and 2 graphs for each. So I began with kind of primitive graphs, and as I learned how to use Python better, I was able to optimize them and make them more clear. And I just made the process a lot more quick as I developed my understanding of Python. And I ultimately ended up with a lot of good results. The final graphs looked kind of like this. These are 2 sample graphs of a lot, 112 graphs. Each of those are single frequency, single sample graphs. These particular ones are different settings on the Aguilar Agro overdrive pedal with the A note, and all together with the different samples with that same note and same pedal, you get that graph that I just brought up. Now that graph is the combination of all of the different settings from one pedal and the same note. So you can kind of see how the sound's been changing with the different settings. I like to point out that 4th harmonic or that 4th spike. You can really see how each one is different. Each setting results in a different height or amplitude, and that shows that the sound has been changed. Now, with all of this data, I had to kind of synthesize it so I could start drawing some conclusions. I began by creating some criteria for a good bass overdrive pedal, and I used the data from the Aguilar Agro and the Boss ODB3, which are both bass overdrive pedals, to come up with some criteria that I could use for defining maybe an overdrive pedal. The first 2 I came up with were open notes, or notes where you're not pressing down on the string. They emphasize that 2nd harmonic. And then for fretted notes, or notes where you're holding the string down, those would be most emphasis on that 1st harmonic. The 3rd would be a steep declining parabolic slope in the frequencies that you would see on that frequency spectra that I showed earlier. The 4th would be the high frequency harmonics would show up when you turn that gain or that overdrive or saturation, whatever distortion setting that you have on the pedal. When you turn that up a lot, you get some higher harmonics. And finally, there are boosts in the amplitude of the first 3 to 5 harmonics, or those are the most great in content. Next, I compared these criteria to the 2 guitar overdrive pedals that I had, the Boss DS1 and the Orange Burst. Now, both of these 2 pedals had good success with the first 2 criterion. They fulfilled those very easily. But the 3rd one is where they had the most failure. They often did not have that desirable steep declining parabolic trend, and often it was shallow or linear or just not very steep. The 4th and 5th criteria were met sometimes by both the pedals, though overall the DS1 was a little bit better. Now, what does all this mean? Well, this data isn't particularly useful for a lot of people. It's a pretty narrow range of relevancy. It's mostly going to be relevant to pedal designers and to musicians. And they're able to kind of look at this data and see what they need to know about each pedal and how well each pedal works. Also, the definition or the criteria that it came up with for the pedal, the bass overdrive pedals are valid potentially for more than just the pedals I tested. And using these criteria and the data that I generated, it kind of confirms that common belief that you really should just be using a bass overdrive pedal with a bass guitar. Now, obviously, music is an art, and you can't just judge music that way. And if someone felt that a guitar overdrive pedal would be best, that's up to them. Now, this project has been very fun for me and a very good growing experience. I'm hoping to continue to learn math at Northwestern University in the fall. I'll be pursuing a degree in computer engineering. I hope to continue to explore mathematics and discover new things and learn new things, as well as study the human voice. That's something that's always drawn my interest, and kind of figure out what makes it unique for each person and how to replicate it. So that is my research project and my presentation. Thank you.
Does anyone have any questions here for Elisha? Very nicely done. Excellent. Okay. It's fascinating to see these places where we get these different big subject areas that intersect, where you're talking about these big differences between how we can analytically look at something like music, which is often so emotional.
Right.
So, excellent. Thank you,
Thank you.
Elisha. So coming up next, speaking of expanding our minds a little bit, I welcome you to expand your own mind as we seek further dimensions, how we can look into other dimensions. I am going to invite up Thomas Chavoda, our computer engineering student, to help explain.
Hi, okay, so before I begin, I do want to let you know that there are a lot of high-level mathematical concepts that we'll be dealing with that I don't have enough time to go into. So if you have questions about what I'm saying, or if there's something that I say that doesn't quite make sense to you, I encourage you, I implore you, please leave me a question in the comments section there. I can access them after I finish with the presentation, and everything will be great, because then I'll get to explain to you, tailored specifically to what you're interested in with my project. So to begin, a few years ago, I became very fascinated with concepts relating to higher dimensional geometry and this fourth dimension, which has a lot of mystique to it in a lot of popular media, but really all it is it's a mathematical concept. Okay. Oh, there we go, figured it out. So in 2 dimensions, something we call 2 dimensions, you need 2 numbers to describe a unique location in space. In 3 dimensions, you need 3 numbers to describe a unique location in that space. And in the 4th dimension, you need 4 numbers to describe a unique location in that space. So it's not too hard of a concept to understand. However, there's issues with us being able to see it. And that ultimately stems from our own human biology, which I'll get into later. So this really perplexed me, and I wanted to be able to see into the 4th dimensions. Anyone would, thinking about this kind of stuff. There have been attempts in the past to try and get around this, try and help us see into this 4th dimension. One attempt is this book called Flatland, which was published in 1884. And it deals with a 2-dimensional square, and he's living in a 2-dimensional world, in a 2-dimensional society. And one day he meets a sphere who's intersecting with his plane of existence. And the sphere can do crazy things like go between rooms and move around and look into different rooms and do all of these things that hurts the square's brain. And so to help the square, this sphere lifts the square out of his own dimension and into the 3rd dimension, where this square's mind is expanded. But there's a problem here. The square's eye would actually produce a 1-dimensional image of his surroundings on his retina. We can imagine that as like the back part of that square's eye there. So the square sees with a 1-dimensional perspective on the world. So being pulled into the 3rd dimension, the square would experience many different confusing images, and he wouldn't really be enlightened, because Abbott kind of expects us to think that the square has a 2-dimensional projection on his eye like we do, but he does not. So this is a problem. That led me to try to wonder, can I remedy this? Can I do something to our vision to help us understand the fourth dimension, like we might be able to for this square, to help them understand a dimension higher than their own? So is it possible to create a four-dimensional visualization program that completely works within a real-time perspective projection simulation? What that means, all that means is trying to get a way for us to be able to maneuver around in the fourth dimension through a simulation. And then what would this visualization look like if we were to do it? So my method mostly consisted of two parts. If I were to include a visualization feature in my code, first it would need to be identified and then justified for its inclusion, just through a logical progression of steps. And only then could I implement it into my code. This was all done through extrapolation and deductive reasoning. Extrapolation is just the way we're comparing the relationship between the two-dimensional to the three-dimensional conversion that we would have to do to the square to our own three-dimensional to four-dimensional conversion of our view. The second part is mostly trial and error. It's going to involve a lot of iteration of code, which is very boring, and I will not show that in this presentation. I will show you the final version that I got to, as well as some tests to confirm that this final version is the best it can possibly be. I did this through the Unity 3D game engine, and this is kind of where my STEM strand comes from. Unity uses C# integrated into it to display things to the screen, so I can use it to simulate fun stuff like the fourth dimension. So to begin with my kind of logical progression through how I came through this visualization, let's start by considering what we would have to do to get this square to see a three-dimensional image. Now, if we can get this square to see a three-dimensional image, we should be able to get a three-dimensional person to see a four-dimensional image. That was my thought process. So the way I figured out to do this was you can cut the two-dimensional image like this, and then you can orient these slices in front of the square's eye, so you still get a one-dimensional view for him, but now he can see lots of different parts, different planes of the three-dimensional view. Now, something to keep in mind here is that the square is actually seeing angled views. So all of these angled views are going to converge on his eye. This is just a top view of actually what's being shown to the square. This is my, this is a still image of my simulation. This is a shape that only exists in the fourth dimension. It's called a hexadecachoron. And this is exactly the same thing we're doing to the square, except we're doing it to us now. So these are angled views that are taking their 3D slices of a four-dimensional scene and a four-dimensional object. So the best way to think about this is to imagine a smooth transition between all of these different slices. So imagine going from one to the other in a smooth fashion. And then you can kind of get a picture of this shape that we're showing to you. So from this, we can deduce that these red dots you're seeing are actually lines in four-dimensional space. And the edges that we're seeing there are actually solid flat shapes, solid faces of this shape that we're looking at. Now, to help understand how we can turn in our space, how we can maneuver around, I developed this thing called the turret model. Let's start with just considering how we as humans can move around and how we can represent that in a computer. So you can think about your direction of the direction you're looking. You can think about that as a vector going away from your eyes, a unit vector. And now this vector can move along a unit sphere of radius 1 around your eyes. You can move around in this direction, and you can move up and down like this. Now, if we were to go past 180 degrees on either side for this up and down movement, we would be confused because we would have, our view would be upside down of the world. It would be very confusing. So a similar thing applies when we add another dimension. These are just cross sections of a hypersphere, a four-dimensional version of a sphere. If we add a third rotation, which we would have to do to be able to see everything on this sphere, we would also have to limit this to 180 degrees. For the same reason we have to limit our view on here. So in four dimensions, we have three possible rotations that we can make: this, and whatever the other one is. Okay. Here's the turret model in pseudocode. All we have to get from this really is that we're using a bunch of trig functions to convert a vector that's represented in memory through angles, and we're converting that to a point in space that we're looking at. From this, we can actually create 4 orthogonal axes that are always relative to the orientation of the camera. And this is going to be vital to getting an accurate hyperplane, just a 3D slice of this 4-dimensional plane that will move when the camera moves. So I derived this equation to help me do this. This is just a combination of a parametric equation for a 4D for a vector, 4D point in space, combined with a dot product so that we can make sure that that point is in the hyperplane. Really what this does is it takes a line segment and a hyperplane and finds an intersection point between them. So we're finding one point along a line segment that intersects with the slice of the 4D space that we're looking through. If we take dot products with this 4-dimensional vector point that we get, and we take a dot product with each of the relative axes that we got from the turret model pseudocode in the last slide, we can actually get a 3-dimensional point that we can plot on the screen. And that's how you're seeing the points with these 2 kind of equations there. And we can actually confirm that this is, in fact, a 4-dimensional view of space by taking a dot product test. Now, this test involves taking a dot product between different orthogonal axes, and as long as the dot product is 0, that means that the axes are orthogonal to each other. They're at 90 degrees to each other. We want all of the axes to be at 90 degrees to each other in order to be a perfect 4-dimensional representation of space. And we can see here that those numbers are extremely small. The largest one you can see there is 1.8 times 10 to the minus 7. That's very small. It's hardly noticeable. And it really comes down to floating point errors done in the calculations that can't be avoided just by how the numbers are represented in code. So I have a video to show you, and this is my code working. You really need to experience this in real time in order to really get the effect of the 4-dimensional view. I'd just like to remind you before I play it is that what you're seeing cross sections. You're seeing slices of a 4-dimensional shape. So just imagine a smooth transition between each of the consecutive slices in order to really get a good accurate picture of what you're actually seeing. The shapes I'm going to be displaying to you are called 4D polytopes. They're extremely symmetrical shapes that have a lot of very cool properties, but they also look very cool when they're shown in this way. So let me play that for you. Right, so a couple of things to go along with this. There are some limitations to the code. There's no way to avoid these slices that we have to take, so we're never going to get a perfect view of the 4th dimension. But I think this is the closest that we can get based on what I've researched. This code is also not optimized, and I can't show anything with a large quantity of points and lines in them. It'll eventually, the frame rate will go so slow that it's difficult to maneuver around. But for low poly shapes, it works just fine. Also, we can't say for certain if this is more intuitive. However, we can say that this is directly extrapolated from a more intuitive way for a square to view a 3-dimensional area. So by that logic, we can then conclude that this should be more doable to see for a 3-dimensional person like myself and you. The implications of this, the 4th dimension is becoming more and more applicable to everyday life. Space-time has been postulated, and this is treating time like another dimension. And when we do this, then space becomes just a 4-dimensional shape. And so we can actually explore this shape with this code. It leads to more visual proofs that could be done with mathematics. And it's generally just could be used as a tool for people to explore this new dimension. So in the future, I intend to pursue similar activities like this and similar research as I go into Cal Poly for computer engineering. I'm really pleased and excited with this result, and I'm really happy that it exceeded all of my expectations for the time constraints that I had. It really did satisfy everything that I was hoping to achieve with this project. I really think that it did achieve the goal of making 4D more intuitive and accessible for everybody. So thank you.
Does anyone have any questions? I mean, probably many questions, but questions that you would like to ask Thomas while we have him up here. He did offer to, if you would like to have more in-depth conversations, I'm sure that we can leave a note in the chat box and we'll connect you guys. In addition, if you feel like you need to see that again, I tell you, having seen it the second time through, I'm doing better. We will be posting the video from our presentations tonight on our school YouTube channel as well. So you'll have a chance to go back through and watch some of this again as well. So, and see any questions. So cool.
Wonderful.
Great, thank you. Nice job.
Thank you.
And now transitioning a little bit from our fourth dimension into the third dimension, we're going to invite up Tyler Werner, who is experimenting with CNC machines and how we can compare one CNC machine to the next. So.
Good evening, everyone. My name is Tyler Werner. Next year, I will be attending Cal Poly for manufacturing engineering. However, I may switch over to mechanical engineering. We need to get my slide up.
Go ahead and click.
Oh,
Sorry.
whoa.
Sorry about
that. So I started researching this because my grandpa had bought a CNC machine over the summer. He spent about $5,000 on this really fancy one, and I thought it was about the coolest thing ever. And I decided I want to build my own. So I started researching it and figuring out how I could do it. And that's what led me to this in my research class. And I decided to develop a method for comparing one CNC machine to another. So my research question was, can I create a proper method for comparing CNC spindle machines? And using this method, how does the quality of the CNC machine I build compare to one of a professionally manufactured CNC machine? So before I dive into my research, I just want to explain a little bit about how CNC machines work. CNC machines work with a set of motors that allow a tool to move around in a 3-dimensional space or 2-dimensional space, even in 5 dimensions too. You can get really complicated CNC machines. However, mine operates in 3 dimensions. You can see on the picture on the right, that is the core of my machine, which houses the spindle, a motor that spins really fast with a carving bit. And the motors around on the outside of the machine move the core around forward, back, left, right, and up and down, which allows the machines to make art or really whatever you wanted out of wood, plastics, or even some softer metals. As you can see here, they operate by removing material from a bigger piece to get the final piece that you want. So here's the CNC machine that I built. This is the mostly printed CNC, or the MP CNC, by the company V1 Engineering. They designed this machine, and the really cool thing about it is all the orange, black, and gold pieces on there were 3D printed at my house, which really lowered the cost of the machine a lot. And then it operates on 1-inch stainless steel tubing. It cost me about $700 to manufacture this machine, and I used exclusively free software on my computer for this machine. And my grandpa's machine, which I compared it to, was about $5,000, as I mentioned earlier, and used exclusively premium software, and he had to pay for that. So when figuring out how I wanted to test one machine to the other, I decided that I need to test it. I could compare accuracy and consistency between the machines. So to test this, I created this test right here. Let's see, there we go, that's fine. I don't know how well you can see it, but I have a positive and a negative shape for each a square, triangle, and circle. I can test accuracy of the machine by measuring the squares and the circles along with their negative hole. And I can also see how accurate they are by taking the positive shape, which theoretically should be the same size as the negative shape, and putting it in there and seeing any sort of observations about it. With a perfect machine, the hole and the positive shape would be the exact same size and would fit perfectly within each other. But since it's not a perfect machine, I can use this to figure out where my machine is lacking or what needs to be improved upon it. In terms of consistency, I would just repeat this over and over again, and the most consistent machine would make the same cut every single time. So I developed this on my computer in AutoCAD. It was really fast to make, and from there sent it to software to make tool paths. You can see the image on the right with all the yellow outlines. Any yellow that's on there will eventually be removed by the machine. From there, I sent them the code to the machines, and I had them cut out twice for each machine. Then I had to prep each of the cutouts. As you can see on the bottom right hand of the screen, there are tabs that were left over on there. The tabs were wood that was left in place intentionally to hold the positive shapes in place. Without the tabs, the wood would have fallen out and might have been altered by the cutting bit a little bit. So I decided by leaving the tabs in and removing them myself, it would create a more accurate piece for my research. I also had to sand them down a little bit because the cutting bit would sometimes leave little bits of wood on the outside of it. From there, I used calipers, which are a measuring device that measures up to 0.001 inches to measure each of my cuts. And I also, from there, analyzed the data and figured out how well each shape fit into their respective hole. And then I used the percent error equation to get a percentage based on that data. So here's my data. I'm not going to go all the way through this just because it's a lot and it's small, but this should be shared with you if you want to look at this more on your own. And then here's the information for my grandpa's machine that I compared it against. So before I talk about my conclusions, I want to talk a little bit about the limitations of my research. So let's go back to the first part of my question. Did I create a proper method for comparing CNC spindle machines? So some of the limitations for this is that I only tested one material, which was this 1/8" plywood. CNC machines, like I mentioned earlier, spindle ones should be able to cut many different types of softwoods, hardwoods, varying sizes, along with other types of materials too. So by only using plywood, I was really limiting the scope of my project. I also only cut in 2 dimensions. I never fully utilized bringing the cutting bit up and down. I really only cut forward, back, and left to right. I also had a small sample size of only 2 cuts per machine, and I only used 1 cutting bit. There's tons of different cutting bits for CNC machines. So by only using the 1, I was also really limiting my sample. The reason I had to limit it was just because of resources and time. I only had so much money to put towards the machines and towards the testing materials for it. And then of course, I only had so much time to get the project over with. So with these limitations, I kind of thought that the method I created wasn't one I would want to publish professionally for other people to use. I feel like if this was retested with more resources available to me, I might be able to refine it and eventually publish it as a real method. But let's just go along with it, and with that question, answer how well my machine compared to the PDJ Pilot Pro. So my machine had a cut deviation of about 0.016 inches to 0.046 inches, which was an accuracy range of 0.8% to 2.3%. And I found that the cuts, both the positive and the negative shapes, they ran a little bit smaller than they were supposed to be. With the PDG Pilot Pro, it had a cut deviation of 0.004 inches to 0.017 inches, which is an accuracy range of 0.2% to 0.85%. And I found that the positive shapes were larger and the negative shapes were cut smaller. In a machine shop, that's actually what you want. You want if you were to cut out a shape and it was larger than the way you wanted it to be, you could always sand it down or remove material to get to the desired size. The same goes for a negative shape or a hole. If you were to cut it smaller than you're supposed to, you could always go back and make the hole bigger. So with my machine, when it's cutting positive shapes smaller than they're supposed to be, that's not a good thing because I can't go back and add material back to it. So that my machine was lacking a little bit, but it's a little unfair to judge a $700 machine to a $5,000 machine. So if I were to revisit this, I would probably judge my machine based on ones that were in a closer price range to it. So thank you for listening to my presentation. If you guys have any questions, that'd be wonderful.
Presentation. Does anyone have any questions for Tyler this evening? Questions for Tyler on his CNC machine? And now you have a CNC machine.
Yeah, I do.
That's awesome.
That's awesome, yeah.
That's pretty great. Any questions out there? All right,
Cool.
thank you very much, Tyler. So we started the evening off talking about the biological science world and looking into our county health lab. Today, we're going to be ending our evening with our final presentation by Natalie Castillo, also exploring the biological realm. This time, though, with some of our favorite insects. Natalie?
Good evening. My name is Natalie Castillo, and I've pursued the health careers strand here at Templeton High School's STEM Academy. I chose to do an AP research project for my strand, in which I investigated the preference of sucrose concentrations of the Vanessa cardui commonly referred to as the painted lady butterfly, in a quantitative study. Now, I had many interests that led to my topic, but one of the first was having taken the course AP Biology in my junior year of high school, taught by Ms. Breckle. In AP Biology, my knowledge was expanded among the basic principles and processes of life, and we got to conduct many experiments. And one of the experiments that we first got to conduct was that of animal behavior. Now, with this experiment, we got to experiment with pill bugs, and we could choose any variable of our choosing to test with. And I thought this was very interesting. And I further loved to learn about the groundbreaking studies with animal behavior, like that of mice, that are present in the medical field today. And this is where I hope to have a career someday. So I thought an animal behavior experiment was the perfect opportunity. Now, I chose to work with butterflies because in our AP Biology course, we were supposed to have done an experiment with butterflies, but due to the COVID-19 pandemic, this was not possible. So I thought this was the perfect opportunity to do this experiment with butterflies and put my own creative spin on it. With that being said, I came up with the following research question. Does the Vanessa cardoi have a preference for higher sucrose concentrations among an 8%, 16%, and 25% sucrose concentration? The following materials would be needed for my quantitative method. 5 painted lady chrysalises, which were bought by Carolina Biological Supply Company, an enclosure that was given to me by our STEM Academy free of charge, a thermometer to ensure that the enclosure was kept at the right or appropriate temperature to raise these painted lady butterflies, which I found to be about 77 degrees, so this worked out perfectly. The sucrose concentrations I would be experimenting with, which I made from cane sugar, purified water, and measuring utensils to create the different ratios for the different concentrations. And I also included a control solution, which would be provided by a 100% water solution. Now, controls are a vital factor in experiments, and this would ensure that my experiment is testing the variable I wanted to test, which was sucrose concentration preference. For example, the butterflies should not have been drinking from this solution as it presents a flaw in my experimental setup or within my butterflies. Now, I would need 4 mesh sponges to hold these solutions and concentrations, and to ensure that there was no cross-contamination of these concentrations, I used napkins. And for the attachment of the chrysalises to the walls of the enclosure, I used tape. In the following image, you can see the package that my chrysalises came in from Carolina Biological Supply Company. This image shows 6 chrysalises, and this was to ensure that I had 5 butterflies successfully emerge from metamorphosis. And this worked out perfectly because unfortunately, one of the chrysalises did not emerge. In the next image, you can see how I went about attaching the butterflies to the walls of the enclosure. In this image, you can see my experimental setup. In the left-hand quadrant at the top, labeled W, was my control solution or 100% water solution. Next to it, labeled 2 teaspoons, was the 8% sucrose concentration. Below it, labeled 4 teaspoons, was the 16% sucrose concentration. And to its left, labeled 2 tablespoons, was the 25% sucrose concentration. Now, from this setup, I generated quantitative numerical data, which can be seen in these 2 data tables. As you can see, data collection lasted for about 20 days, and data collection was done by periodical observations throughout the day because I did not have a camera to oversee the butterflies' movements 24/7. In the top row, demonstrated in the blue color, is the control solution or 100% water solution. And this demonstrates the distribution of butterflies feeding on a concentration throughout Earth in a total amount of each day. Below it, in the red color, is the 8% sucrose concentration. Next, in the yellow color, is a 16% sucrose concentration. And finally, in the last row, in a green color, is the 25% sucrose concentration. Now, from this data, I generated a line graph in Google Spreadsheets, and I thought this was a perfect way to analyze my data. And from this line graph, we can see that the blue line, or the 100% sucrose constant, 100% water solution, did not have any butterflies feeding from it. As you can see, that means that my control acted properly as a control, and that my experiment was testing sucrose concentration preference. Another pattern I would like to point out in my line graph is that of the 25% sucrose concentration, which has an overwhelmingly dominance of the others in this graph. Now, in order to statistically support this, I used what is called a chi-squared test. I came across this in my AP biology class and thought it was a perfect method for statistical support of my generated data. The chi-squared test has an equation that looks like the following. The chi-squared value is given from the sum of observed frequencies minus the expected frequencies squared all over the expected frequency. Now, before I could do this calculation, I needed what was called a null hypothesis. My null hypothesis is there is no statistical difference from the observed frequencies and expected frequencies. Now, in order to accept or reject this null hypothesis, I needed the degrees of freedom and critical value. The degrees of freedom are the number of possible outcomes of the experiment, which I concluded to be 3 because of the 3 sucrose concentrations, minus 1, which gives you a degree of freedom of 2. Now, this degree of freedom yielded a critical value of 9.210, and if this critical value was above my chi-squared value, then I would have to accept this null hypothesis. However, if the critical value was below my chi-squared value, then I would get to reject this null hypothesis and fully conclude that there was a statistical difference in my data that can be inferred from the variable tested sucrose concentration preference. As you can see, my chi-squared value was 11.23, which was bigger than the critical value. Therefore, I was able to reject this null hypothesis. So the observed frequencies you see in each of the calculations shows that this distribution was indeed from the variable being tested sucrose concentration preference. Now, I would like to address the limitations of my experiment because I think it is important to do so. I narrowed down my limitations to 3 factors, which I thought were time, sample size, and a lack of equipment. I say time because I think had I been given more time, I would have been able to do much more trials and had the time to care for a bigger sample size, which was another one of my limitations. And I think having 10 times the amount of what I worked with, because I only worked with 5 painted lady butterflies, would have given me much more sufficient data that would have led to much more credible analysis. And finally, I say a lack of equipment because I didn't have a camera to observe the butterflies 24/7, which I think is a critical aspect of this experiment. So therefore, I would recommend more research into this field, and if someone or myself were to return to this experiment, that they would respect these limitations and make changes accordingly. Now, I plan to attend UC Davis in the fall to further my education and research in majoring in biological sciences. I definitely have a passion for this kind of work, and I'm so excited to be carrying on my education and to do more experiments like these at UC Davis. Thank you.
Does anyone have any questions for Natalie? Any questions out there in internet land? I love seeing how we can analyze our insect behavior and see how that applies then to other scenarios. I know you had mentioned an interest in going into the medical field, and by understanding animals in different ways, we can understand ourselves better too.
Yeah, especially.
Yeah, so it's a nice tie-in right there. Well, I'd like to, before we end the evening, I'd like to invite up our whole cohort up here. All 7 of our students up here, proud on you right here. I am so proud of these students right here, and I hope that you've had a chance this evening to see some of the amazing work that they are doing out here. So we want to say thank you to all of you out in internet land for joining us here, and all of you up here. We can give a big wave to all of your family members out there. Thank you so much.
Hi, Mom.
Thank you for joining us. That wraps up our program this evening. We look forward to continuing on this program and following along with what some of these great scholars are doing here in the future as well. So thank you very much. You all have an excellent evening. Yay.